Okay, I guess we are live. Welcome everybody to this talk of the Sussex Vision. My name is Marion Silies. I'm a PhD in the lab of Tom Baden at Sussex University. And before I will start to introduce our guest today, I would like to thank the organizers of the Worldwide Neuro Initiative, especially Tim Vogels from the University of Oxford, for providing the general framework which makes it possible to have these talks online. So thank you very much. Today, we are delighted to have Marion Silies on the program. She's not only my previous supervisor, but she's also one of the leading experts in the field of Drosophila genetics and vision. She started her science career by doing a PhD at the University of Münster, which she finished in 2009. After that, she moved to the University of Stanford and did a postdoc with Thomas Clandini. Here she studied the motion vision circuit in the Drosophila optic lobe. And after that, she moved to Göttingen, where she became a PI at the European Neuroscience Institute. And she continued to study vision in Drosophila. And I guess the spectrum became a bit broader, so a bit more vision in general. And since 2019, she's a full professor at the University of Mainz. I'm really happy to have her today, and I guess most of the work that she's presenting will be from the time in Göttingen. Before we start the talk, I would like to say some things about the organization. So if you have any question at any time, you can post that in the live chat, which you can find either right next to the video or maybe under the video. And also, if you have questions that we will not answer after the video, then you are happy to join our Zoom meeting after the video or after the presentation. We will share the link in the live stream after the talk is finished and after we have answered some questions. Yeah, and I guess it would be fun to just have a casual hanging out after the talk. I guess that's everything I wanted to say. I hope I haven't forgotten anything. So yeah, please welcome Marion Silies.
Okay, thank you very much, Marvin. So let me share my screen with you. Okay, so I would, of course, like to thank Marvin and the Baden Lab for having me today, and yeah, all of Worldwide Neuro for making this possible and adding so much fun science to the last weeks. So I'm very excited to share with you what my lab has been doing over the past year or a few years, and I'll talk about how vision can work in dynamically changing environments, and more specifically, how visual systems can accurately compute contrast in rapidly changing visual contexts. And so I will start by showing you a movie that my previous graduate student Katja took of her dog Yuppie. So what you see here is Yuppie catching his toy. Yeah, and what you can appreciate is Yuppie is very good at doing that. And he can do that sort of like regardless of what the external light conditions are. And we actually learned this by going on one of the very first lab trips. We went onto a hike where we sort of got lost in the Göttingen woods, and Yuppie was with us, and we sort of like entertained him by throwing a ball. And while we could like hardly see our path anymore, like Yuppie was still like, you know, very constantly good at like catching his toy. And as you can see here, and he can even do that when you are sort of confounding the experiment by shining like a bright light at him, so that Katja could take this movie. So, and if I explain this to you, then that visual processing works efficiently in very different light conditions, then you will probably all think of adaptation. Yeah. So the reason why visual systems can like handle such different light conditions is that they are sensitive to contrast, or more specifically temporal Weber contrast in this case. And sort of what visual systems are sensitive to is the difference in luminance between object and background normalized to background. So because of this term here, vision works sort of very stably across different adapted states and therefore different light conditions. And this is a concept that was also very beautifully introduced by Jeff Diamond's talk that I hope you all had a chance to see here on the Sussex Vision channel 2 weeks ago. But there is a problem, or there might be a problem for visual systems when the background is quickly changing. So here I just drew a fake tree and a fake shade into this image here. And so I think there's a problem when Yuppie will try to catch a ball that we are throwing into the shade for him. And this is because the visual system is not adapting instantaneously. So adaptation is very fast, but it doesn't happen within milliseconds. And this is shown here in like older data, for example, from the Retina. So this is a scaled Retina here on the left-hand side. So you see that if you sort of like measure adaptation, there's a fast component to adaptation that happens maybe even within tens of milliseconds and seconds. And then here there's a very slow component of adaptation that lasts up to seconds. And these different time scales of adaptation are true in both invertebrate and vertebrate visual systems and also in humans. So here are some human psychophysics experiments where you see that there's a fast adapting component in the response that works on the order of hundreds of milliseconds, and then there's a slow component that lasts tens of seconds. And so the problem is that if you, or if a visual system encounters a visual signal sort of like within where background luminance is very quickly changing, then contrast-sensitive neurons won't be able to compute Weber contrast correctly. And this is something that I'm showing you in this term down here. So here, so basically what a contrast-sensitive neuron would then be sensitive to, so it would still sense the luminance difference between the object and the background. So in this case, like the dog's toy and the shade of the tree, but it would sort of like divide it by the luminance of the background. So in this case, the bright snow. And this would lead to a wrong estimation of contrast, so to a too small contrast response. And this is something that we sometimes call apparent contrast. And this is, of course, not only a challenge for dog visual systems, but for visual systems in general. So my lab uses fruit flies, Drosophila, as a model to study visual processing. And you can like easily imagine that if a fruit fly is trying to escape a predator, like the praying mantis here, then for the behavior of the animal, it would be advantageous if it was able to like do this equally well under all of these conditions. So in bright daylight on the left, at dusk or dawn, or when it's, for example, when the predator is hiding in a shade, for example, under a leaf here. Yeah. And again, the problem here is that what adaptation can achieve is contrast computation in slowly changing environments. So, for example, the contrast that the fly is calculating, sort of looking at the head of the praying mantis here, in bright background will be the same. So the contrast signal will be the same at dusk or dawn because of adaptation. But if adaptation hasn't hit, then the challenge will be to compute sort of the luminance difference between this object and the background divided by a too large background from the previously adapted state. So contrast sensitivity might underestimate a stimulus in quickly changing environments. And today I will, and again, so what I'm like sort of illustrating in gray here is that it would be advantageous for the fly to exhibit contrast constant behavior, to have equal behavior responses to visual signals under any of these conditions. And today I'll tell you about some work that we did over the past years, and I'll show you that luminance information is still present in the visual system and ensures the accurate estimation of contrast in dynamically changing visual environments. And a part of this work was actually published earlier this year, so you can read about it later if you want to hear more. And in the published work, we basically showed that luminance can like sort of do exactly what I just showed you, namely fix this discrepancy between contrast-sensitive responses that sort of underestimate the visual stimulus and the actual behavioral response. But then I'll also show you unpublished data showing you scenarios where the opposite is true. So I'll show you like some data where we found conditions where contrast signals would overestimate the relevance of a visual stimulus. And I will show you that luminance information can also fix this. And if I have time, I will also talk about unpublished work looking at the cellular and circuit mechanisms of this luminance-based scaling of contrast computation. And I would just like to add here that there's a lot of data in here that I've actually never talked about before. So it's a bit scary to do this in this context for the first time. But this also means that I would really appreciate your questions and feedback and criticisms. So I'm looking forward to many questions afterwards, and also to a lively discussion afterwards for those of you who are interested. Okay, so let me start by telling you about our system. So again, we work with Drosophila. This is a Drosophila eye. It's composed of these 800 single units or ommatidia. And downstream of these 800 ommatidia, you will find 800 units. So basically, you have 800 columns that together form a retinotopic image of the outside world in each eye. And if we take a cross section through the system, then we basically see the structure illustrated here. So here on the outside, which I didn't show you, is the retina, where the photoreceptors are housed, and then photoreceptor terminals are coming down into this first structure here, which is called the lamina. And there they synapse onto a series of so-called laminar monopolar cells, or large monopolar cells called LMCs. And they are L1, L2, and L3, and you will hear a lot about L2 and L3 today. And these are the inputs to pathways that are specialized to detect contrast increments or on stimuli and contrast decrements or off stimuli. And L2 and L3 are two inputs to the off pathway. And they synapse on a series of interneurons here, which then in turn project onto the dendrites of these direction selective cells. So here, down here in the lobular plate are the first direction selective cells of the on and the off pathways, and they are called T4 and T5 in Drosophila. And what we know, so we basically know extremely well how these peripheral stages of visual processing respond to light and to contrast. So they have been like extremely well characterized by the labs of Simon Laughlin, Roger Hardy, Mikuyu Sola, and others. And so how a photoreceptor responds to light looks like is shown here in this orange box. So basically a photoreceptor will respond to a step change in luminance with this like transient response that basically already signals contrast, and then a sustained component, which is luminance sensitive. And then all these laminar neurons downstream, or the laminar neurons downstream, or together the LMCs are described to be amplified, more transient, and sign-inverted versions compared to their photoreceptor input. And this is something that you can see here. So you see that they have the opposite sign signal in response to light. And this is because photoreceptors release histamine, and then all the LMCs will express a histamine-gated chloride channel. So you have a sign-inverting synapse at this stage in visual processing. And then you see that these LMCs here, and these are, I should maybe already add that these are mostly data from L1 and L2 neurons here, are really transient versions of the photoreceptor input. And the idea is that they are like very contrast sensitive, and that the visual system sort of like got rid of luminance information at this point. And so what we asked, or what we wondered, if these contrast sensitive responses like this early in visual processing are fully sufficient to explain behavioral responses to contrast under dynamically changing light conditions. And to ask this, we basically did behavioral experiments. So we put a fly on an air-crushed ball. It's situated in an arena in which we just can display visual stimuli. And then we showed the flies stimuli where we had a dark edge moving onto different background luminances. And sort of every time we like show a new moving edge to the fly, we show a new background luminance. And what is important is that all of these stimuli are of minus 100% Weber contrast. Yeah, because it's a dark edge, the luminance will be zero, almost zero. And this means that Weber contrast will always be -1 or minus 100%. Whereas the apparent contrast, so like the contrast response of a non-adapted cell, will increase with increasing background luminance. And so we basically asked, how do flies respond to these different minus 100% Weber contrast stimuli, and how does this scale with contrast-sensitive responses in laminar neurons? And to get at the contrast-sensitive responses in laminar neurons, we could go back to, again, all of these recordings that have been done in these cells, and they have been so extensively characterized that we could basically predict the responses of these contrast-sensitive responses based on existing literature. So, and this is all work done by a really brilliant PhD student in my lab, Madura. And so what Madura did, she first measured behavioral responses to these different off edges. And what you can see is that the behavioral responses here really scale with Weber contrast. So again, these are all minus 100% contrast stimuli. And what you can see here is that the behavioral responses are not significantly different to each other. So the flies can respond well to each of these off edges, even when the background luminance is quickly changing. Okay, and then she compared this, or she first predicted the responses of these contrast-sensitive LMCs based on existing data, and basically found that contrast-sensitive LMCs would have a smaller response to responses onto a dark background. And if you compare this with the behavioral responses that Madura measured, so this is from the same data as shown on the previous slide, only here I'm plotting the peak turning velocity only to the 5 different edges here, then you see that there's a discrepancy where a contrast-sensitive response alone cannot account for a behavioral response if the background just turned dim, so in sort of like contextual dim light, yeah? And this is basically illustrating the problem that I showed you in the schematic drawings in the beginning, that under the conditions where the background just turned dark, you basically have a contrast-sensitive response that would underestimate the behavioral relevance of the stimulus. And so what we hypothesized is that there must be a corrective signal that sort of like accounts for this discrepancy and ensures appropriate behavioral responses to contrast, even in dynamically changing visual environments. So what could be accounting for this discrepancy? So we got some insights from work that another PhD student, Katja, was doing. So Katja is a PhD student who recently graduated and just actually sadly left the lab 3 weeks ago. And Katja did very extensive characterizations of these lamina cells that I introduced to you already. So Katja expressed the genetically encoded calcium indicator, GCAMP, in either L2 or L3. And I should just remind you that whenever I say L2, I mean L2 as a cell type. So there are 800 L2s in the fly visual system, 1 in each of these 800 ommatidia that we have. So basically, this is how this looks like. So Katja is recording calcium signals from the exon terminals of L2, and you see that they are in one layer in the visual system, there are several exon terminals lined up next to each other, and the same for L3. And when Katja shows these flies like very simple light flashes, then what we see is that there's an increase in calcium signal in response to off in both L2 and L3 neurons. And whereas the calcium signal in L2 goes back to baseline fairly quickly, the calcium signal in L3 is really sustained. Yeah, and we thought this was interesting for several reasons. One of them is that very similar properties have been described in the virtual retina. This is actually data from Tom Baden from a bit more than 10 years ago, where when he was like doing a very similar experiments recording calcium signals in bipolar cell exon terminals, he also saw these like transient and sustained bipolar cell types. And we also thought that this is like interesting with respect to the idea that we need this like corrective signal, but that all lamina neurons were thought to be contrast sensitive. So we decided to look at this carefully and designed a stimulus that sort of allows us to really distinguish contrast and luminance sensitivity. And this is inspired by a paper from Jeff Diamond's lab, where he like discovered that there's luminance sensitivity present in the virtual retina in A2 emigrant cells, for instance. So what we did is we adapted flies to a bright background for 30 seconds. And actually, I should say that this is something that Marvin did when he was a master student in my lab. So what Marvin did, he adapted flies to a bright background for 30 seconds. And then he showed them 2 sequential off steps, the A and the B step. And each of them can take on one of 6 different variants. And so the A step will always differ in both luminance and contrast, but then the B step, and this is the important one, is designed so that it will take on one of these 6 different luminance values, but the B step will always be 25% contrast relative to the A step. So this really allows us to distinguish a contrast from a luminance sensitive cell. Namely, a contrast sensitive cell will respond with equal amplitude to all of these 6 different 25% contrast steps. And here in blue, I'm showing you the responses of these transient L2 neurons. And when you plot the responses, the calcium signals as a function of contrast here, then what you see if you look at these 6 different data points for minus 25% contrast, you see, well, maybe you can't even see, but they are all lying on top of each other. Yeah, so this is really how a contrast sensitive cell looks like. If we instead plot this as a function of luminance, you see how similar they are. Okay, if we do the same for L3, then we see that this looks very different. So the responses to these 6 different 25% off steps already differs. And what you see is then if we plot the responses to the stimulus as a function of luminance, then L3 calcium signals respond very similarly to nearby luminances, no matter if they were coming from the A or the B steps. So this hints towards L3 being a luminance sensitive cell. We tested this explicitly by sort of doing the opposite experiment where we fix luminance. So we allowed random transition between 5 different luminances, and these steps are then associated with like different contrasts. And what you see is even in like responses of single L2 exon terminals is that L2 neurons have this like very, very flat baseline. And there's this transient response in L2 neurons whenever there's an off step, and this transient response is larger, the larger the amplitude of the step. This looks very different for L3. So L3 basically like doesn't have like one baseline really, but there's a constant change in like steady state whenever there's a step change in luminance. And what you see here is that even for like, you know, 2 or equal steps, so 2 equal step changes in luminance, L3 responses can be very different. So here you see like a large increase in calcium, and here you basically see no response. But what instead L3 appears to be sensitive to is like really the luminance of the stimulus. So for example, here in black, I'm showing you all the transitions to complete darkness on the screen. And this is always associated with the same calcium signal or the same calcium steady state response in L3 neurons. And so we can like pool all of these responses. So this would be the response to like any of these steps landing on complete off. And if we do this also for all the other steps, then we see that L3 has this nonlinear decline in response amplitude and with increasing luminance. Yeah, so it's a luminance sensitive cell that is active in dim light. Yeah, and this is basically perfect. This is exactly what we need to like fix contrast computation. So we hypothesized that luminance information from this L3 pathway might provide this corrective signal that ensures these equal responses to the same Weber contrast stimuli. So that ensures contrast constancy. We tested this by going back to behavioral experiments. And so again, the idea, so what I showed you before is this discrepancy between contrast-sensitive neuronal responses and behavior. Yeah, and if we are correct and this L3 pathway provides sort of like this corrective signal here, then taking out the L3 pathway, and this is something that we can genetically do in flies, should sort of like bring these behavioral responses down or closer to these contrast-sensitive responses. So this is what we did. This is what we tested. So we genetically blocked the L3 pathway. And first I'm showing you wild type, so our control behavioral responses again. This is essentially the same data that you have seen before. Only now I'm sort of like not plotting them on top of each other, but on the left-hand side, you see the turning responses to off edges moving onto dark backgrounds, in the middle intermediate backgrounds, and then on the right-hand side, bright backgrounds. And again, you can see that the behavioral responses to these different, off edges moving onto different backgrounds is essentially the same under for all of these stimuli. If we now block the output of the contrast-sensitive pathway L2, you see that L2 is required for all of these, for behavioral responses to all of these conditions. So what I'm actually plotting here is the turning response as a function of time. And in gray is the time where the off edge is sort of moving across the visual field of the fly. And what you see is that the turning response is strongly reduced in all of these conditions. So the contrast-sensitive pathway is required under any of these conditions. When we block the outputs of the luminance-sensitive L3 neuron instead, what we see is that the peak turning amplitude is not altered if the off edge is running, or if the fly is turning to an off edge running onto a bright background. We start to see in phenotype and intermediate background luminances, and you see that the turning response is severely altered when the edge is running onto a dark background. Okay, if I summarize these data and I plot them together onto this like curve that I showed you before, then you see that this behavior response of L3 silenced flies now looks much more similar to contrast sensitive responses alone. And what this basically shows is that luminance information is really like the thing that enables proper contrast responses, and this is provided by the system. We are this like parallel dedicated pathway that gets inputs from L3 and that is sensitive to luminance. Okay, but so far, I only showed this sort of like for one adaptation state. Yeah, so this fly is sitting there. It's like encountering these like randomly interleave 5 different edges. But in principle, when we, the visual system is like able to encounter or will encounter many, a much larger range of luminances. Yeah. So how would this like basically compare to if we like put the fly into, dusk or dawn or like near darkness or something? So this is something that we tested. So we characterized these behavioral responses at many different adaptation regimes. And we did this by using the same set of five stimuli, where of edges are running onto different backgrounds. And then we changed the whole range of luminances that the fly was encountering by adding neutral density filters to this. And by doing this, we could cover 4 orders of log magnitude as shown here. And what you see here on top for wild type turning is basically almost a continuous function of luminance. And then we measured behavior down to a regime where like basically both the flies and actually also us humans can't see anything anymore. So until we are like reaching sensitivity threshold down here. And then we asked, okay, so what's the requirement of this luminance sensitive pathway under all of these conditions? And how does this again scale with signals that would be provided by purely contrast sensitive first order interneurons? So Madura again went and predicted contrast sensitive responses for all of these conditions. And for any of these adaptation states indicated by one grayscale here, you see that contrast sensitive responses would always underestimate a visual stimulus in dim light relative to the range of luminances that the fly is adapted to. Yeah, and this is something that we call contextual dim light. And then when we compare this with like the behavior of flies in which we block L3, then you see that for any of these conditions, this L3 block behavior is actually very similar to these contrast sensitive predictions. Yeah, so for any of these conditions, for any adaptation state, L3 is required in contextual dim light. So dim light relative to the range of luminances that the fly is adapted to again. Okay. And yeah, so this means that L3 can like basically fix contrast computation over many different adaptation states and over a large range of like absolute luminances. And something that we also saw in the data, and I think we didn't point out in the paper, but I'll point this out today, is that when you look at like the responses that are nearing this like, dim end of where flies can actually like still see anything, so like low absolute luminances, what you see is that it appears to be that L3 silencing also has an absolute effect. So basically here for these adaptation, like these 2 dimmest, these 2 experiments where we use the most ND filters, so where we have the lowest absolute luminances, what you see is that L3 is not only required, in contextual dim light, but it's required overall. So here you see that L3 silence behavior doesn't match wild type behavior even on the bright end of this adapted state here. This is something that I come back to in a bit. Okay. So so this is all published, and so what I hope I could convince you so far is that luminance information is required for this accurate estimation of contrast when the world is quickly changing. And so I illustrated the problem to you that you have like 2 small contrast sensitive responses if the world is just like quickly going dim, and this can be corrected by this luminance signal that we identified. Okay, but so far. I only talked about like sort of, you can say, a very special case, namely 100% contrast. So a condition where like complete dark edges are moving onto like different backgrounds. So what about other contrasts? So Madura continued to characterize behavior and showed different contrasts by having a not completely dark edge running onto different luminances, and these would be associated with Weber contrast ranging from 50% to 87%. And what you see here when in the wild type turning responses here is that the behavioral responses again scale beautifully with Weber contrast. So the larger the contrast, the larger the response. And so we then look more closely at these 50% contrast responses, where you see that wild type turning is actually very low. And we design stimuli that are now all 50% 50% Weber contrast, so minus 50% Weber contrast, but are associated with different foreground and background luminances. But again, all of them are 50% Weber contrast. And again, what you see is when you measure behavioral responses to any of these stimuli is that they essentially look the same. So behavioral responses, again, scale with Weber contrast. If you have different ways to achieve the same Weber contrast, you will still have the same behavioral response. And so then we ask what happens to these behavioral responses if you now take out the luminance sensitive pathway. And what we saw is that under these conditions, so when you now block A3 here, you actually see an increased behavioral response. Yeah. And what this basically means is that this is like, and I mean, maybe just so I'm not showing you like these LMC predictions here, but just so you can like even grasp this intuitively. What you are seeing here is a very, very small wild type turning response to these contrasts, right? But 50% is half of 100% contrast. So contrast sensitive LMCs will still have like a strong contrast sensitive response to, a strong response to like any of these stimuli, but apparently the behavioral relevance of these signals is very low. So you have like very low wild type turning in response to these signals. And this is again something that you sort of like uncover when you silence L3. So the idea is again that like L3 sort of like matches the behavioral relevance of or the discrepancy between the behavioral relevance of a stimulus and a purely contrast sensitive behavior. Only under these conditions, it sort of like is doing the opposite. So luminance information here suppresses behavior if a purely contrast sensitive response would overestimate a visual stimulus. Okay, and then Madura didn't only do this for one adaptation state, but also did this for many different adaptation states, again, by using ND filters. And what you see here is that this is, again, something that holds true for different adaptation regimes. Yeah, so here on the right-hand side of this plot, you again see that L3 or the luminance sensitive pathway via L3 suppresses behavioral responses to very small contrast. And again, just note how similar, all of these wild type responses in gray are to all of these 50% Weber contrasts. Yeah, and again, this is something that that you don't see anymore when you silence L3. So you sort of need this luminance sensitive pathway to keep behavioral responses to Weber contrast constant. And what you also see is that there seems to be like a switching point on the left-hand side of this plot, where you turn the behavioral phenotype of these L3 blocked flies from having enhanced responses to having lower responses. And again, this is something where L3 appears to have a role in absolute dim light. And this is the same dichotomy that I pointed out earlier for the minus 100% Weber contrast data, where I put these errors and said, look, there also appears to be an absolute contribution there. And that is something that you see much more clearly now that we have this overestimation case otherwise within the data. And so this seemed complicated to us. So we thought, okay, there are apparently like many different things that this luminance sensitive pathway is doing. It can fix overestimation and underestimation of contrast in contextual luminance conditions, but then it also has this absolute requirement. So we went to collaborate with Shuai Zhao, a graduate student in Juliana Georgieva's lab at the Max Planck Institute for Brain Research in Frankfurt, and discussed the data with them. And I should maybe say that it was really Shuai who pointed out this dichotomy in our data and showed us very clearly that this is there when he's just subtracting the two behavioral conditions, so control and L3 silencing. And then, this was even before we had these data for low contrast, where this is becoming very evident. And so based on all these observations, he devised a model and trained this on the data. And so we basically now have one model that really can capture all of these components that we see in our data. So that sort of explains or that shows this like underestimation of visual signals when L3 is silenced for high contrast, the overestimation for low contrast, and also the absolute requirement in like absolute dim light. Okay. Okay, so what I showed you so far is that luminance information is basically doing many things, and sort of like the overarching theme is that it ensures the accurate estimation of contrast in the dynamically changing visual environments. And for the last 10 or 15 minutes or so, I will tell you about the work that we are doing to figure out how this is done. So we are like interested to understand the cellular and circuit mechanisms of this luminance-based scaling of contrast computation. And there are many open questions out there, and one of them is like, how is contrast constancy achieved in visual circuitry? And this is a question, the project that Burak Gur, another graduate student in the lab, took on. And so far, I only showed you data sort of comparing these contrast-sensitive responses in the very periphery, so in these first-order laminar interneurons, to behavior. But ultimately, you know, and I showed you that L2 and L3, so the two input neurons here have very different sensitivities. So one is sensitive to contrast, the other one is sensitive to luminance. And the question is like, really, where are these like two pathways, or where's information from these two pathways combined? So this clearly has to be achieved somewhere pre-motor. And so what we know is that these neurons project onto different neurons in the medulla here, and they really have sort of like their own target neurons in the medulla, but then information converges latest at the level of the dendrites of these direction selective T5 neurons. So we thought that this would be a good start to check. And what we now did is we used exactly the same stimuli that we used in behavior and showed them to flies while we recorded calcium signals in these neurons here. And what you see here, so we now recorded calcium signals in these direction selective T5 cells for exactly these, you know, sort of like stimuli that I showed you, where we interleave these 5 different background luminances, and then we run off edges across the fly. We match them to the directional preference of these direction selective cells here. And then what you see is that T5 neurons are contrast constant. So these off direction selective cells exhibit contrast constancy already and show sort of like invariant responses or statistically non-significant responses to these different minus 100% Weber contrast conditions. And again, so far when I talked about contrast sensitive responses up here in the lamina, we generated these predictions from previous data, but here we decided it's about time that we like record these neurons ourselves and look at calcium signals under exactly the conditions that we use in behavior. And what you see here is basically what I told you before, is that L2 neurons themselves don't exhibit this contrast constancy. So they still have this problem that they have like a too low calcium response or too low neuronal response in response to contrasts for like these large dark background luminances. Okay, so that means that, you know, this combination of contrast and luminance information must happen at the latest when information is coming together onto the dendrites of T5 cells, and we are currently investigating if this could potentially already happen before that in presynaptic circuitry. And then the next question is that we spend quite a bit of time on is the question how contrast constancy how contrast and luminance sensitive pathways become specialized to be so different to begin with, yeah, because these are just two first-order interneurons that are sensitive to completely different features of the visual scene. How can this be the case? And this is again a lot of work that Katja did, and she now teamed up with Junaid Akhtar, a postdoc in the lab who is coming from functional genomics. And so we first, our first hypothesis is that maybe it's already in the photoreceptor input that these two neurons are getting. Because, I mean, the idea is that these motion detection circuits get predominant input from R1 through R6 photoreceptors, but there's also data that report that sort of photoreceptors, which are people more often think are more associated with color vision, like R8, can also contribute to these circuits, and that R8 might be feeding into L3 neurons. Yeah, and so we thought, okay, maybe it's true that sort of like less adapting R8 cells contribute to this sustained component in photoreceptors. And we did a series of experiments to show that this is not the case. So, for example, here's just one of them. So we know that these outer and inner photoreceptors, so R1 through 6 or R8, express different opsins and rhodopsins. So we basically tested the spectral efficiency or the spectral tuning of L2 and L3 neurons and used this as an indirect measurement to ask where from which photoreceptor they get the input. We did this by building LED synths in collaboration with Gregor Belusic and his student Marco, where we basically take a series of LEDs and send the light via a diffraction grating and through a lens onto the eye of the fly and can like sort of very precisely show them many different wavelengths. And when we do this, then we see that the spectral tuning properties of L2 and L3 are really like fairly similar and or both peak at the point where RH1 has its peak sensitivity. We see some difference in the UV range, but this is not relevant for what we have been looking at here. So, and we also did a lot of genetic experiments where we sort of isolated the different photoreceptor components. And I can just tell you that L2 and L3 both rely on R1 through R6 input predominantly. So then the question is, I think, an even more interesting one. So how do you, sort of like separate these two signals at sort of just one synapse between these photoreceptors and either L2 or L3? And to just sort of know what sort of like calcium signal we'd be dealing at the photoreceptor terminals, Katja recorded responses from the photoreceptor terminals here. And you can see that the calcium signal, so the steady state response in photoreceptors also tracks luminance, but sort of has a more linear relationship with luminance than L3 neurons do have. And what you also see is that the photoreceptor calcium signal increases when the light is turned on. This is the opposite of these laminar neuron responses, but this is what I already explained to you, and this is due to these histamine-gated chloride channels. Okay, so we are sort of dealing with two types of transformations here, yeah? So on the one hand side, we need to like transform this like sort of linear relationship to luminance that is present in the photoreceptor axon terminals to this nonlinear preference for dark in L3 neurons, and then we completely have to eliminate this luminance sensitive baseline in L2 to get these purely contrast sensitive responses out. And so we asked, and there are several possibilities how this can be achieved, and maybe very coarsely, this can be sort of like cell autonomous mechanisms, or this can be done through circuit interactions. So to distinguish the two, we started to look at L2 first and recorded calcium signals not only at the axon terminal, but also in input regions and along the axon. And the idea here was to like really see how the dendritic signal is that L2s are, inheriting or are getting from photoreceptors, or how they respond to a photoreceptor signals at the postsynaptic site. And then to see if there are any transformations happening along the neuron, which would argue for circuit, or could argue for circuit interaction. So this is something that we actually saw in L2. So we saw that the dendritic calcium signal is actually not as transient as the calcium signal at the presynapse, arguing that some transformations happen along the neurons. I just said that, of course, these can also be cell intrinsic properties, but this is something that we then further tested, and we can test this by genetically isolating L2 from its circuit environment. And this we can do by taking ORT mutants. ORT is encoding the histamine-gated chloride channel. And so basically in an ORT mutant, no one should be talking to photoreceptors. And then we can use fly genetics to put this channel selectively back only in L2 neurons. So the only synapse that is intact here is the L2 the photoreceptor to L2 synapse. Again, so what I showed you before is that when we show these like different steps of luminance, or these different luminances to flies expressing GCAMP in L2, you see these like one fixed baseline, and you see these like purely contrast sensitive transient responses in L2 controls. When you do the same experiment and you record from L2 neurons in this L2 ORT rescue conditions, so again, this is like a situation where L2 is sort of or where the photoreceptor L2 synapse is the only one that's really present. And what we see here is that they still have this like transient contrast sensitive response, but they also start to get this luminance sensitive plateau component. And what this argues is that the sort of like elimination of this luminance sensitive plateau component between photoreceptors and L2 neurons depends on circuit interactions. And then we did the same for L3 neurons. So we again recorded calcium signals in L3 from the dendrites, the axon and the axon terminates. And here, in contrast to L2, the responses basically look the same no matter where you check. So dendritic calcium signals are sustained. We don't see any transient component in the response there. So this argues that further difference between L2 and L3 are likely mediated by cell autonomous mechanisms. To get at these, we Junite, the postdoc in the lab, started to do some functional genomic experiment and isolated RNA from like either L2 or L3 neurons. We actually use GCAMs, so we used exactly the same experimental conditions that we use for imaging for sorting, and then we obtained cell-type-specific RNA sequencing datasets. I should also say that this is something that was previously done in Larry Tsipersky's lab, but during development, we initially looked at these data sets and then decided that it makes sense to us to redo that in adult flies. And then what we did initially, we thought, okay, now we just, sort of like look at all the molecule classes that, can mediate these like different properties, so channels, transporters, anything that has to do with synapses, and look at those. And we screened like many different candidates there. So I think Katja and Burak tested a whole of, 50 or so different candidate transporters and channels that are differentially expressed. And we didn't find anything that's like really strikingly changing the properties from a contrast to a luminance sensitive cell or vice versa. So what Burak found is, some like sort of milder phenotypes. So for example, this is data where Burak found that two potassium channels, Shaker and Schall, are preferentially expressed in L2 neurons. And when you take them out, in this case, using RNAi, but you also use pharmacology to do that, then you see that you are sort of like changing the temporal dynamics a little bit, and you make them a little bit more sustained. Yeah. But again, what we were hoping for is like really genes that really differentiate these like, contrast sensitive L2 from these luminance sensitive L3 neurons. And so after, not finding anything in like, a lot of screening, we thought, okay, maybe there isn't like a single gene, like a single channel or something that like really is alone mediating these responses, which I think makes a lot of sense. So to still get at these differences, we decided to look a little bit like more upstream, molecularly speaking now. And instead of like looking at, synaptic genes, we looked at transcription factors that could specify a whole cohort of proteins that could then like mediate L2 or L3 properties. And what we found, or what we first looked at, is a transcription factor that had already been known to be L3 specific. It's called DFAS-FORM. And during development, it was already shown to be basically only expressed in L3, both at the RNA level and at the protein level. And when we looked in our adult RNA sequencing data set, we see that so here, these are like the pink shaded areas are the exons, is that they are also expressed in L3 in adults, but not in L2. And so DFAS-F was shown to be required for the cell-specific layering of L2 and L3 during development. And we thought to ask if it maybe also has the function in mediating the physiological properties of L2 and L3 neurons. And I should say that this is was done in very close interaction with the lab of Matt Picott, who was like really a fantastic person to have around and discuss science with and exchange like tools and ideas, who unfortunately lost his battle to cancer like a few months ago. And so the images that I'm showing you here were generated by Ivan Santiago, a PhD student, or now graduated postdoc in the Picott lab. Okay, so what we are doing here is we are basically looking at individual clones L3 neurons that are dominantly labeled with GCAMP. And so what you are seeing here is when you record from the exon terminals of these individual clones, then you see again how sustained L3 neurons are and how they track luminance. Actually, in this genetic context, so this is a fly that has a lot of transgenes, they are a little bit more spiky than they normally are, but these responses are still totally luminance sensitive. So these are L3 controls. If we now mutate these neurons to lose a DFSF, so this is now a dominantly marked homozygous mutant DFSF clone in an otherwise heterozygous background, then what you see is that these DFSF mutant L3 neurons have very different physiological properties. So they sort of like become spiky and lose this like shift in baseline. So they sort of like start to have one baseline and really start to resemble L2 responses. And this is something that we can quantify across like many cells in many flies here. So they really start to look more like L2 neurons here and have this like flat plateau response. And if we explicitly test contrast and luminance sensitivity using this A/B step stimulus that Marvin brought to the lab, what Katja, who now did the experiment, found is that you really turn a previously luminance sensitive neuron into a contrast sensitive L3 neuron. And this is shown here. So these are control data where I showed you before that L3 controls show very similar responses to nearby luminances, regardless of whether this was the A or the B step. This is no longer true in DFSF mutant L3s. And you see how very similar or indistinguishable the responses to these 6 different 25% contrasts become in a DFSF mutant. Okay, so to summarize this part, we are sort of on our way to working out the different signal transformations that happen in the lamina or at the photoreceptor to lamina synapse. And what I showed you is that a large part of the difference between L3 and L2 neurons is established in a DFSF dependent manner, and that the elimination of this baseline in L2, in normal L2s, is further dependent on circuit interactions. And with this, I'm almost coming to the end. So I just want to highlight that the challenges I introduced today are, of course, not just the challenge for like the fly visual system, but are a common challenge for visual systems in general. So, you know, I like illustrated this by showing you 2 examples of dogs catching balls and flies trying to escape predators. And again, I just want to like highlight data from Jeff Diamond's lab, who again spoke here 2 weeks ago, that showed that luminance information is also present in the vertebral retina postsynaptic of photoreceptors. So they showed that there's a transient component in, for example, A2 amacrine cells that is sensitive to contrast, and a sustained component that like really scales with luminance. So I think it's really an appealing hypothesis to like think that this luminance information could be there to ensure contrast constancy in many different visual systems. So to summarize, I showed you that luminance information ensures the accurate estimation of contrast in dynamically changing visual environments, and that this luminance sensitive pathway can adjust for both overestimation of contrast, which is new, and I just showed this for the first time today, and for the underestimation of contrast, which is something that we previously published. And then I talked about the cellular and circuit mechanisms of this luminance-based scaling of contrast computation. I showed you that within a circuitry, this happens presynaptic or onto the dendrites of these direction selective cells. I told you that the luminance information from contrast-sensitive L2s is eliminated in a circuit-dependent way, and that the DFSF transcription factor is required to mediate these physiological L3 properties. And with this, I again want to like highlight or thank all of the people that have done this work. I think I mentioned Burak and Katja and Marvin and Madura throughout my talk. I unfortunately didn't have the time to talk about the work of other students in the lab, like Miriam and Luis and Juan and Sebastian, maybe some other time. And I also just really want to thank our excellent technical support, so Christine, Jonas, and Simone, for their assistance. I think I mentioned our collaborators, and I just want to thank my colleagues at the University of Mainz. So this is like a really newly forming neuroscience department, and it's a lot of fun to be here. And so thank you all for the great first year here. And I also want to, I should probably acknowledge funding. So we were funded by the Eminota program of the DFG Collaborative Research Center in Göttingen, in ERC, and now by the University of Mainz. And I also want to say that we are hiring postdocs and PhD students. If you are interested, we are now located in the beautiful city of Mainz in a newly built biocenter. And again, I think this is a great place to do neuroscience. So shoot me an email or contact me else if you are interested in this. Okay, thanks for your attention. I'm happy to take questions.
Thanks for the talk, Marion. It's a really nice story, I would say. I think we have about 50 minutes to take questions. People have a bit shy on asking questions so far. But I was thinking about something that maybe also is interesting for other people. So when you speak about these turning behaviors, maybe you could explain a bit more what the actual behavioral background is, because normally flies are not sitting on a ball and seeing kind of bars moving in front of their eyes, I would say.
Let me, sorry, let me just seeing you, so I see who I actually talk to. Okay, so yeah, I mean, this is like clearly a very simplified setup for like, you know, showing like something where, I mean, I talk about like dynamically changing visual scenes and stuff. And I mean, everything that we have been doing is like show like sort of boring edges to fly. So this is like true. What is true is that this is like, I think, a nice system to illustrate that, you know, how behavior responses scale with contrast. And yeah, so I mean, I think it would be super interesting like to bring this into like more real world scene statistics soon, and maybe start to look at natural scenes and stuff. And this is something we are like definitely interested in doing in the future. I think so far, you know, I think it was, I mean, I hope I could convince you that we could like learn something from like using these, you know, very simple visual stimuli to do this. But I think now is the time to start and look into this using more natural stimuli. Yeah.
There's one question, is if the transient responses that you are seeing, if they are all stochastic, which means like suggesting there is a spiking process underlying, or perhaps vesicular release variability, or are they all always the same?
Okay, so what I didn't say is that these are non-spiking neurons. So, you know, very similar to the periphery in the vertebrate visual system. These are all neurons with graded potentials. So, yeah, so what we are basically seeing is, what we are measuring is like the presynaptic calcium, which will like turn into a release of neurotransmitter, and this, you know, sort of will give graded information about the input that these cells are getting. And so in terms of stochasticity, I mean, these are like really robust responses, I would say. I mean, I showed you data for individual exon terminals in which you can like basically really see how the responses scale with contrast. It's fairly easy to record from these neurons, and, you know, sort of like just by looking at these like single cell traces, even you can like really see that they scale with contrast, you know, if they have a luminance sensitive plateau or not. What we typically do is we record from many neurons at the same time. So we can like, and usually like record 8 to 10 exon terminals in one fly, and then also across average across many flies, but you generally get a really good sense by just looking at single neuronal responses.
Yeah. The other question is if there's any evidence for axonic inputs at the L2 terminals, and do you think that that this is a source of differential response you observe to L2 cells?
Yeah, so this is a very good question. I mean, it's definitely, so what we have in the fly visual system, we have EM0 reconstructions that basically, you know, really like tell us which neurons are there and who is talking to whom, and by how many neurons these cells are connected to each other. There are, you know, always like some axonic inputs as well. You know, they also get like a lot of inputs in their dendritic regions. There are some reciprocal synapses in the terminals even. So I think there's like a lot of room for like circuit inputs there. What we have done so far, maybe I just talk a little bit more about this question. So what we have done so far is that we have sort of, I mean, we wanted to understand where, like who's basically like, you know, responsible for this like elimination of this luminance sensitive baseline in L2 neurons. Yeah. So, and the most obvious candidate seemed to us that this could like be coming from L3 neurons, because L3 have like a luminance sensitive component. So, and they don't make direct synapses, but you know, there are some like interneurons, GABAergic interneurons also, where we thought, okay, you know, this like would be relatively straightforward to feed the signal back onto L2 neurons. And we have imaged L2 neurons while we silence L3 neurons, and we don't see any change there. So it doesn't seem to be coming from there. So that's something where we still don't know if there's maybe someone else who like is grasping photoreceptor input and then like feeding this back, this information back onto L2 to eliminate this baseline or something. So this is something we are still investigating. There are a few more candidates that we can test based on these EM data. Yeah, and again, nothing we have done so far. I'm not sure it has to be exoaxonic. I mean, it could totally be dendritic, I think. Yeah. So, and then you can like have some cell intrinsic properties that also do this further. To see where this baseline sort of is eliminated is something, you know, we would have liked to do. So, I mean, this is something we like didn't manage to achieve by recording the dendritic signals for these stimuli as well. It's like really hard to get to the dendrites because they are sort of like, you know, just hidden underneath the retina. I mean, the axon terminals of these neurons, you know, sort of like project to the next neuropil in the medulla. It's really easy to get there. But to perform calcium imaging experiments sort of like from the lamina itself is very tricky. So Katja had to try very hard and like manage to get these full field flash responses, but it's impossible to show like many stimuli there and get conclusive signals, responses there. So this, unfortunately, we can't answer.
There's another question. Do you think that the L3 neurons are slower because they are luminance sensitive, or is this just a coincidence?
Yeah, so I'm not 100% sure what you mean with slower. So increase, like the initial increase. Well, I mean, if you're talking about the initial increase in calcium, then that is something that we see for some stimuli. That is true. So well observed in the data, I would say then. And where this is coming from is something I can't fully answer. I can tell you that this is something that we were actually messing a lot with when we were modulating individual channels. So when we like, you know, sort of like trying to find something that is changing luminance sensitive L3 response into a contrast sensitive L3 response by taking out individual channels, we would often affect sort of like just the kinetics of this initial onset response. So that seems to be a cell intrinsic property. And there are actually many different channels that sort of have this phenotype when you knock them out, where you're sort of messing with the initial kinetics. But yeah.
I think the question was if L3 neurons are slower in their responses than L2 in general.
Yeah, so I think they are a bit, it's nothing we have looked into very carefully. What I should also say, I mean, you know, there will already be some people who are like, all you're doing is just measure calcium, and how can you talk about fast or slow when you're just measuring calcium? So what we also did is we, and it's in the paper, so we also thought to look at this like very initial fast response in both of these cells. Because, for example, what you might have noticed that in our calcium recordings and photoreceptors, for example, we only see this like luminance sensitive plateau, and we don't see like the fast, you know, contrast sensitive signal that I showed you in the voltage recordings from photoreceptors earlier. So we wondered if L3 neurons, for example, might actually also have like a fast contrast sensitive component that we have been missing whenever we talk about this luminance sensitive calcium signal. And so what we did is we expressed ASAP2, so a voltage, genetically encoded voltage indicator in both L2 and L3 neurons. And if we do this, then in L3, we also like see a very fast contrast sensitive like spike, not spike, but fast response basically. And sort of like the functional relevance for that was then that we wondered if, you know, so L3 like is basically L3 just a pathway that scales contrast computation in other pathways, or can it itself have like a more active role in motion computation, for example, by having a fast contrast signal itself that we sort of have been missing in these calcium recordings. And so we try to get at that functionally by asking if we only have L3 neurons present, which we can do in these odd rescue experiments that I explained to you, then is this like sufficient to respond to motion? And it is. So if you sort of like have a visual system where you have only photoreceptors talking to L3, you can actually get behavioral responses to motion. So you don't need these like dedicated contrast sensitive pathways for that. And what is also interesting is that this is like something that is specifically prominent, like in contextual dim light again, which is then totally matched with this other function of L3 that you are like need this pathway to do well in these like specific luminance regimes.
Okay, maybe one last question. And this is something, of course, that we also try to investigate. I guess I know back from the time where I was in the lab, but what's with this hypothesis that L3 gets less input from photoreceptors than L2? Do you think that has anything to do with the difference in behavior?
So this is really the question that Marvin should answer, because this was his master thesis that we thought, okay, you know, maybe it's this is like the reason that they sort of like saturate earlier, and this like makes up these differences. And I guess Marvin will agree with me that we like couldn't find like any strong evidence for that.
It's also quite tricky to do. It's also quite tricky to do. Like, it's
true. So what we were like messing with is like, you know, sort of like the amount of receptor on the postsynaptic side. You know, there are some ideas on how you can like actually change how many like synapses a neuron is making onto a postsynaptic cell, which is then hard to validate. But sort of like with the different like genetic tricks that we tried, we didn't find anything. So I wouldn't say we can say it's not the reason. I would say we at least so far have tried and haven't found evidence for it. But this is a very interesting question. I'm actually also, maybe I should add to that, I'm not 100% sure that the idea is that like L3 is providing many fewer synapses into like these pathways actually holds true if you start to consider potential wide field neurons. Yeah, so I mean, like, yes, how many synapses does photoreceptors make onto L3 and L3 onto their downstream neurons? But then, you know, there are all these uncharacterized wide field neurons which could have a very interesting role for this like contrast constancy idea. So we will see.
Okay, I guess I thank you again for answering all the questions and for giving the talk. And to everybody who's still watching, if you want to ask more questions to Marion, you can join us on the Zoom. I will just post the link now. And yeah, feel free to join us. Yeah, I guess that's it.
Thank you.
So I keep the live stream open because if people want to see the link,
okay,
the link will disappear. I managed
to look at the questions myself. I was sort of Mike. Okay. I should have checked to see who is asking a question.
Yeah, maybe I should have read the names. It was a bit challenging to read and ask.
Answer some more questions meanwhile. So if people are still listening, I can. Terufumi asked, at 50% Weber contrast, there was a behavioral response at the stimulus offset. Where can the behavioral response come from? Yeah, this is totally true. It's something that we see. And we don't know exactly where this is coming from. It for sure sort of like depends on the adaptation state of the fly. So it's something that we can alter if we change the interstimulus interval. So for everything that I've showed you, we keep sort of like an interstimulus interval at 1 second where it's completely dark. And if we don't do that, but we sort of like use any other interstimulus interval, we lose that response.
There was one more question. On top of the autonomous function of L cells, do you think that the different input is also important for the difference between contrast luminescence function? Not really sure.
On top of the cell autonomous, do you think the different inputs are also important for the difference between contrast and luminescence function? Yeah, for sure. I mean, this circuit input that L2 is getting, if we eliminate that, they are not sensitive to, they start to become sensitive to luminance as well. So that is something that is for sure also contributing there. Yeah. you're all right. It's very hard to read and answer questions at the same time.
Then I think the last question that it says, do you think that this contrast insensitivity could extend well to other types of behavior? The S cells may have specialized circuits for computing motion.
The S cells? Ach, die S cells.
The S cells.
Marvin, can you read this question once more?
So do you think that this contrast insensitivity could extend well to other types of behavior?
So I'm not 100% sure I understand the question, because what I was trying to convince you that the behavior is very contrast sensitive. Yeah. So even if contrast sensitive neurons alone can't handle that, and that you sort of have a mechanism to fix that. So
maybe the question would be like, if you silence L3 and then you try different behavioral tasks.
Yeah, so this I definitely think will be the case. I mean, I think, you know, there are many behaviors which, visual behaviors which sort of like scale with contrast, or which initially rely on contrast. So I'd be very surprised if that was like a phenomenon that's only there for motion-guided behaviors. It's the only thing we've tested so far, because it seemed like very easy, and there are these like very well-characterized responses to motion. Yeah, so. Tom Baden asked, is it possible to measure the actual release from L2 and L3? Yes, that should be possible, and we haven't done that yet. It would be really interesting, because what we are very interested in is like sort of to see the time scale for which you have this luminance-based scaling, right? Because at some point adaptation will catch up. So how are these two balanced? Yeah, so when adaptation catches up, you shouldn't like have an L3 response anymore. And we were trying to get at this by recording calcium signals in L3 neurons. And our idea was that, you know, maybe naively, that their response will just go down at the time scales at which like adaptation goes up, and we don't see any of that. So and it's unclear if that just, you know, really is true that L3 neurons aren't adapting, or if that's something we just haven't seen in calcium imaging. So we are very much interested in measuring release. Actually, now that I say that, I remember that we tried once, and I don't think it's like as beautiful as, you know, what as beautifully possible as in other systems. So we have very high baseline expression of so for example synaptofluorins, that gives us very tiny delta F over F, so that makes it a bit tricky, but now that we have like all these indicators for that we can express on postsynaptic sites to look for glutamate and acetylcholine and stuff, that should be possible. So both L2 and L3 are cholinergic, so if we could potentially, so this is like an experiment that we could definitely do and measure acetylcholine release, basically.
Yes, apparently they're working now quite well. I don't know if you saw the talk by Gautam Avatramani, so he's gotten that to work in the mouse.
The acetylcholine sensors, or?
Yeah.
Okay.
So maybe there's room for that to work. That would be great.
Yeah, and we have them and stuff. We just haven't done the experiment, so.
Yeah.
I think those were all the questions from the YouTube.
Yep.
I will keep bugging you though. So, you know, in vertebrates, we're quite obsessed about counting.
And now I see everyone. Hey, Michael. Hey, Beth. Hi, Simon.
So, you know, in the vertebrates, we're really obsessed with counting cell types, and then we're giving them names, and then, but I guess in flies, you're a little bit further with this, right? So.
Luminance sensitive, so these are L3 controls. If we now mutate these.
Oh, that's Simon, I think.
That's someone's YouTube is still on.
Dominantly marked homozygous mutant DFSF clone in another. That's horrible. I hear my own voice.
Maybe I think it's Simon. Maybe you could mute
very different
your YouTube.
They sort of like become spiky and lose.
Wait, I can.
Maybe
I can mute.
You have to take control.
Yeah, I wonder how you.
Probably. Yeah, just mute. Ah, good.
It's something that we can quantify across like many cells.
Okay, no, sorry.
Okay. Yeah, so sorry. So, so what's the situation in the flies in terms of cell types? So how many L, is there an L1, L5, L7? Where does it end?
It ends at 5. And L1, 2, and 3 are the ones that are getting direct input from photoreceptors. So.
Okay.
Yeah.
Manageable.
It's kind of weird, right? That the other ones, like L4 and L5, are still weird, right? Like,
right.
Like evolutionarily, they're weird. Like, why are they in the lamina in the first place? when they're basically medulla neurons, right? They're just weird.
Yeah. Simon looks skeptical.
Muted him, so I can't say anything at the moment.
So Marion, can I ask
Yeah.
again just about timing? Because it's like my own personal obsession. So this is in no way like a shortcoming of your beautiful work, but I just I've always been interested in like the like at the limit, right? When it seems like the thing that makes vision so cool is that it's actually useful on the time scale of milliseconds, right? Especially for like escape behaviors. Right. And so I just wonder, it's not obvious to me which is harder, right? To make an accurate contrast estimate or an accurate luminance estimate. They both actually seem kind of hard, right? If you have, 50 milliseconds, something is sweeping by and you've got to do it, right? It just seems weird to me that L3 just seems slower, right? At steady state, it's definitely slower, right? It can't track changes. And in our original Lamina paper, when we silenced L3, like the deficits we saw were on slow stuff. So that's why in my head, I just have in mind, like, L3 is a slow neuron. It mediates slow vision. It's not obvious. Does it make sense though? Like,
yeah, I mean, so yes and no. So I mean, I agree that it's like, so I mean, yeah, it maybe doesn't track like fast changes as much. What we have seen is, for example, how long does it take to get to steady state in L2 and L3 is exactly the same. I mean, yeah, you guys have like looked at this more carefully, where you guys have shown that it's required for slow motion. So that would be something that you would be looking for in this like contrast sensitive component in L3, right? So, which is something that we basically don't see in calcium. So I can't really answer this question like at the timescales that you are interested in, I think. So then we would like have to do voltage imaging for all of these, or?
Right. No, I think you can still do. I mean, I think if you just do like, I mean, this is if I were like, to do follow-up experiments on your experiments in my lab, which I won't do, don't worry. I would just do everything over like 2 or 3 orders of magnitude in time scale, right? Just make all the stimuli super fast. You can still look at the, slower calcium response to a fast event, right? That's not a problem.
So you would just be looking at the filters and see how they look like.
It just seems like the hardest thing about vision is not the steady state stuff, right? It's like doing things over like super short time.
But the steady state stuff is doing cool stuff, like.
Yes. Right.
So, yeah. No, I agree with you.
No, but your protocol is like, it's a cool mix of the steady state and the transient, right? Because it's just this edge sweeping through, right? It pushes on both.
Yeah, it's true. Yeah. So I mean, I can't answer. I mean, we actually like really never like, measured filters or like very fast step responses of these neurons. So maybe we should really add that to like the standard step of stimuli when we are testing more like, of our, I mean, we still like hope to get at the mechanisms that are downstream of this DFSF transcription factor. And now that we have like this handle, sort of go back to the membrane and maybe, modulate a few things where we know that they are now there. I mean, we like sort of, did this RNA sequencing now also in DFSF mutant clones, so we can like go back. So maybe when we like test candidates there, we'll like include that. And so maybe I can answer this question next time. Yeah.
Is it easier to ask questions about timing with the behavior? It'd be really cool to do, maybe you've already done different speed sweeps, but across these.
True, you should like, I mean, that's a good idea because that's something that Michael could pull out in the Tuttle study, right? Like these differences in like requirement for different speeds for L2 versus L3. So that would be interesting. Yeah, it's not so, as you know, it's not so easy to screen a lot of candidates doing behavior. I mean, and then look at speed tuning, right? I mean, the beautiful thing about these like calcium responses is that they are so robust. Yeah, so there you just look at a few exon terminals and you know if you have a phenotype or you don't. So yeah. Really but now that I have armies of like undergraduates here in Mainz, I can do that.
Can you say something else about, do you have like, so for going for targets downstream of a transcription factor, that's presumably like, I don't know, hundreds of genes or something, or many dozens?
Yeah.
Like, do you have, is the idea to go for like top candidates? Like,
so what we did is so we have still things that are normally differentially expressed between L2 and L3. And now we did RNA-seq between L3 neurons with or without DFSF. So we know which of the genes are differentially regulated in these neurons by DFSF. We are currently complemented this with ChIP-seq to just like see who is like a direct target. And then what we did is we sort of like grouped them functionally. So now, we are still interested in like voltage gated channels and channels in general and transporters. And now we sort of like have clusters of potassium channels where we see, okay, you know, there's like 6, 3 of them are upregulated in L3 and 3 are upregulated in L2. And generally the ones that are in L3 are more of the Ka type or something like that. So, you know, that I think gives us a better handle. So we are like, yeah, so there are like a few C and G channels, which would make a lot of sense for like having these very sustained responses and stuff. So that's what we are now looking into, to sort of like see, based on these large lists, are there like groups of genes where they could like function together and make sense? And then, yeah, I don't know. It will be hard to get there, right? I mean, I don't know, even with flip-stop and stuff, like to do a quadruple flip-stop potassium channels or something. We'll see.
But it also just seems like we love, right? Our methods are best if there was like 1 or 2 channels that explain huge effects. We're totally set up for that. But if it's like, you know, 15 channels together, like how do you do that experiment?
So we actually tried to do the opposite, but haven't succeeded so far. This is like maybe the craziest idea I've like ever followed. I'm not sure I should tell you before we even start to do these experiments, but I tell you anyways. So like, you know, what I was thinking is that this might be a very cool test bed for like, you know, sort of like Eve Marder type of ideas where you have these like huge parameter of like channels that like mediate certain properties. So what we were thinking, now that everyone is doing single cell sequencing to get at cell types, what we should do, we should like do single cell sequencing only of L2 neurons. And so sort of like see what are sort of like the functional clusters that you get within a certain cell type. And, you know, maybe, I don't know, can you get at the depths of yeah, sort of like, and it's actually something that wasn't only inspired by like, you know, us not being able to figure out, you know, what is like mediating these differences, but also by the fact, if you, for example, for these, you know, Shawl and Shaker things, when we either do pharmacology or we do RNAi, it's really like you see a phenotype or you don't see a phenotype. And this is completely different from fly to fly. So the variability there is much higher than for like any other gene. So if we like take out DFSF out of L3 neurons, they are always contrast sensitive. If we do the same for any of these, we now in the lab call them terminal genes, yeah, then they might or might not be required, which makes a lot of sense if you like look at Eve's work, right? So that's like sort of maybe an experimental system in which you can actually get at this sort of variability.
I'm always. Oh, hi everybody. I'm sorry I was late joining in. I had a lot of trouble with my computer. I always go back to the fact that at least in our hands in Califera, it looked as if the on transients and off transients in L1 and L2 were produced by modulating the chloride current, the postsynaptic chloride current in L1 and L2. And this was done by measuring conductances and reversing the potentials in intracellular recordings. It's a really robust result. And the puzzle, and that what that suggests is that the background subtraction, which isolates contrast, is being done by either inactivating progressively the histamine-gated chloride channels or by regulating transmitter release from the presynaptic terminal. Now, the interesting thing is that L3, L1, and L2 share the same neurotransmitter at tetradic synapses. So if I was going to look for something in L3, I would look for something about something that would modulate, for example, its ORT receptor. I thought the ORT receptor experiment was really cool, by the way. I really like that.
Thank you. Yeah, so the answer to this is that Marvin really should have stayed in the lab, because this was exactly like what he was like after when he started, you know, that we also thought, I mean, what we did is we like played with like these ORT receptors, we overexpressed them, see, you know, do levels, do something. And then we said, okay, what we really should do is like look for things that interact with this. I think you are absolutely right. So maybe that's also like a good idea to like look into this again when we are now like scanning our candidates to sort of see, you know, what could potentially interact with these histamine-gated chloride channels. You're absolutely right. Yeah, that's very good, valuable comment.
I also agreed with Michael's comment. I think you should try some other stimuli to get behavioral responses and to get physiological responses, because these transients are just dreadful things. They're incredibly non-linear, particularly in monopolar cells, and they've got all sorts of different components in.
Yeah.
And so it's really difficult to interpret what's going on.
Yeah.
The deeper you dig, the more confusing it all becomes. And in the end, I gave up.
But with these transients, if you gave up, I'm not sure I'm going to try. But so like, I mean, with these transients, you mean like these like very fast on transients, for example, that you only see when you measure voltage, right? Or
yes. Well, I mean, it's partly that, but it's partly the fact that your on and off isolating stimuli, which are receding edges, which actually cover the whole receptive field. The hedge goes down all the way down the receptive field, and on one side it's white and on the other side it's black or gray. They are very unusual stimuli in space and time, and they may do, you may get some quite weird behavioral responses to that, not to say physiological responses.
Yeah.
And because they're wide field, they tend to confound changes in spatial receptive field with all the other changes.
Because who's wide field?
Sorry?
Who is wide field?
Your stimulus is very wide field.
Yeah.
And we know that at very low light levels, there's spatial pooling in the lamina of signals.
I think that is exactly the reason why we see this dichotomy, you know, where we start to see.
I think that could be it. And so there's some different mode of processing going on at really low light levels.
Yeah, I fully
agree.
agree.
It's very interesting in its own right.
Yeah. I mean, what we are definitely doing at the moment is to try this with more local stimuli and also see, you know, under which, I mean, sort of how wide the spatial pooling is. And I mean, like really the idea would be that you, I mean, also like, you know, what you don't want to is you don't want to use luminance information to just like even out all contrast computation. That would also, you know, then you basically like,
no.
Yeah. So what you want is you want like something that like where your luminance sort of based scaling is a little bit more global, right? So, and that's, I hope something that we can get it by using like stimuli of different sizes, and then maybe also, and this is, I mentioned the DM neurons like at the end, right? So they could be like really the circuit implementation of this, where you have these dorsal medulla neurons that span several columns. So there you could like combine signals from several L3s to like use that to compute contrast constancy basically, and then you still keep your local contrast signals from L2.
Yeah.
Madhuva, I see that you are there. And
all these things are possible. All these things are possible. The trick is to design an experiment which can tease them apart. And that's a big challenge. But when it works, it's fun.
I agree.
I mean, can I just suggest like a fun, I mean, one of the challenges with the behavioral silencing experiments is for the earlier neurons, like the monopolar cells, it's hard to interpret and even make use of a particularly subtle phenotype, right? So I think you revert to pretty, you know, like things like just large luminance contrast changes, maybe a little speed tuning, right? You can try a few classes of stimuli, but it would be hard to do much with more subtle things. Like an experiment that I think would be really beautiful is find some, if you wanted to get away from motion detection, right? Find like a different output pathway, like a looming sensitive neuron, which is pretty close to the kind of stimulus, right, that Marion is already showing, right? But the readout is turning, so then do a looming sensitive, and then ask how that, you know, feature detecting neuron, like say like an L4 or an L6 or something, is modulated by manipulations to L3. Like that would be very much in line with the kind of stimulus regime you're already working on. You can imagine flies really want to be able to detect looming stimuli at all ranges of, you know, luminance without the like very particular effect of, you know, things which are optimized for motion processing, right? Like, because otherwise I think just using behavior alone, it's going to be quite hard. I think if you're like, you know, mutating a channel in an L3, right? Like that's going to be very hard to read out.
Yeah, I mean, I think for all of these things, I was more thinking of physiology, but yeah, then it's hard to get at this very fast component. So yeah, I think it's a good idea.
I think Norma got a question.
Norma, you wanted to say something?
Yeah, I wanted to ask something because you showed these like really nice like attenuation of the on responses in the L2 cells. I think it was an attenuation. So basically, like in the beginning, you have like a response to the off, but also to the on, and then the on response attenuates, right? Do you know anything like why that happens, or like is there some kind of inhibition in these very upper layers.
What do you mean with attenuates?
Like, so the response is there, but then it kind of flattens out.
So, I mean, why do these L2 neurons have transient responses in the first place, or?
No. More like, more that you basically have a transient response to the on, but like later on, it's not there anymore, right? So
how do these cells become rectified, or?
Yes.
Well, I mean, like, so L2 cells are like, well, somewhat linear. So, you know, this has been like sort of like fairly extensively looked at, and like sort of real rectification in these on and off pathways is happening like a synapse further down. Yeah, so the reason why you sort of like see only a tiny on response, like sort of negative, you know, like negative calcium signal there, or decreasing calcium might partially be because of like calcium indicators and low baselines in these neurons, and yeah, partially that there's already some nonlinearity within these cells themselves. But yeah. Does someone else want to add something to that?
Okay. So basically, it's not really known what makes them like because they are linear in the beginning, right? But then like the on response kind of is.
I mean, rectification you often get like sort of for free at synapses. Yeah, I mean the question is, I think oftentimes more how do you keep a system linear? So if you want to do this, like
but I mean like maybe I'm also reading the plot wrong, but it looks more like actually at the synapse it's still like non-rectified, and then it becomes rectified. Like wow.
So all of the L2 data I showed like all at the exon terminals. So you're meaning like when I like dendrites exon terminals. Yeah, so I mean there I think, well, actually I'm not 100% sure how well you can tell from this what is actually like the on response or like a non-complete decay to off yet or something. So that's, yeah, so maybe then that one would have to do with like more like an intermediate gray. I mean here these are just full feed flashes, so I think from there it's very hard to talk about rectification, so I don't know.
Okay.
Yeah.
Thanks.
Really nice talk. I don't have any specific questions. I just wanted to say hi.
Hello, Yvette. Congratulations for your awesome job.
Oh, thank you.
Congratulations, Yvette.
And I guess for almost getting married last weekend.
Oh yeah, I know. It's so crazy. It's been good though. We still celebrated. We made a cake. Yeah. And nice to meet you, Madura. Very nice data.
Same here. I saw it for the first time. Only heard about you so far.
So this is Madura who did, I mean, I'm pointing where Madura is on my screen, but it's different for you. So this is Madura who did all the LMC predictions and behavioral experiments.
That's beautiful.
Yeah.
Okay.
Yeah. Okay, I actually have to run, but I wanted to say hi. Fantastic talk. I really enjoyed it.
Thank you.
Bye, everyone.
Bye.
Bye. Bye.
Okay, any other questions, comments? Yes, a few people which don't show their videos. Natalia, do you have something to say?
Natalia is the good spirit of our lab.
Okay.
Hello, Marion. It was a great talk, actually. Yeah, I'm not like I'm not showing off today, so it's okay next time. but thank you for your talk, and thank you to Marvin for hosting. Yeah, it's actually nice. I mean, I haven't met most of you people, so it's nice to see some faces. Maybe one day mine.
Okay, it's nice to hear your voice, Natalia.
Thank you so much. Bye-bye.
Okay, Tom disappeared. Marvin, you should try these things in the WordPress Retina.
Oh, yeah, first I have to get my own recordings done. No, but I guess with the MEA, it's much more easy to get the fast responses. So, yeah. Yeah, I mean, I guess if there are no further questions, then.
Well, I'd just like to point out one thing. You probably know this anyway. Sorry to jump in. But in the Hardy and Vekstrom paper, which is the only paper which really discriminates between L1 and L2 and L3 using intracellular responses and where they mark the cells, it's a really fantastically accomplished paper. as you'd expect from Roger. And in there, they found very consistently that L3 always had a larger plateau response than L1 and L2, which fits in very nicely with what you're saying. It's exactly what you would predict if L3 had a bigger luminance component.
It's absolutely true. And, you know, I mean, they also like, you know, sort of assigned different like potassium conductances to these neurons. So all of that makes sense. And actually you even see that like in like, you know, these, which paper, like Usitola, like, you know, sort of like some of the like previous papers where you like sort of, you know, see L3 type neurons in other species. You actually, if it's like never described, but if you look at the voltage traces and you sort of like draw a baseline in there, you always see that like even the L3 voltage response never returns completely to baseline. Yeah, so I actually had like a backup slide where I was like going to show all the like, you know, previous examples where people had like assigned L2 and L3 types to their data. And it's always there. Like, you know, it's not that we found something that like wasn't in the data before. It's just, you know, but I mean, they definitely like in voltage recordings always have these fast contrast sensitive peak, which is like clearly dominating these responses. But yeah, it's not something that we are now reinventing in Drosophilites. It has always been there in the data. Yeah.
But there's always this big problem. I think we discussed this before, that there's this big extracellular potential in the lamina that's induced by light. So you never really know what the baseline is. And really the only baseline that counts is the baseline that's seen by the presynaptic terminal of the monopolar cell in the medulla. And that's in a different electrical compartment. Yeah. So this is a really interesting, tricky problem.
Yeah.
Because you actually don't know what the membrane potential is. And when you correct for what is your best estimate of the membrane potential in L1 and L2 recorded in the lamina by subtracting away your best estimate of the extracellular bias potential, you find there is quite a reasonable plateau component.
Ah, okay.
So that's in the, if you look in the Vekstrom-Laughlin paper from 2010, it's in there.
Right, so it's a good thing that
we
are
don't
recording.
make a big deal of this because they're all sort of dirty secrets all coming out, you know. Sort of stuff we don't know that we ought to.
I'm glad this is all consistent with your previous recordings then. And I guess, you know, it's one of the selling points for measuring presynaptic calcium then, because we are past all of these like potentials in the lamina.
Yeah,
so Simon won't go there,
but that's a skeptical old-fashioned voltage person, you know. So.
But as you point out, there's not like one voltage, right? You'd like voltage in all compartments, and you'd like all the references. So, you know, one electrode is also a imperfect measurement, right?
Yeah.
All right.
Okay.
I mean, I don't want to cut this off, but I think
I'm ready for a cup of tea.
If they're not,
okay. Thank you all for coming.
Yeah, thanks for the talk, Marion.
Thank you.
Yeah.
And thank you.
Thank you.
Very interesting talk.
Thank you. Bye-bye.
Bye.
Okay, bye.